My name is Marley Romero. Today is March 11, 2018, and I am doing the aseptic technique for the cultures for this week's assignment. We had to culture the E. coli, the S. epidermidis, and then the yeast. I'm waiting for the yeast to activate and start kind of bubbling, you could say. And once that happens, then I'm gonna collect that one and put it into my test tube that I have here, where they will grow over the next 48 hours. And what I did first was wipe down my countertop, remove everything that I'm not gonna be using or that I don't want to get contaminated or possible exposed to any of the stuff I'm culturing. I do have my candle lit right here, as you can tell, and I was reading over the instructions a few times just to make sure I understood what to do prior to collecting the cultures. I'm still waiting on my yeast to activate. I have like about maybe 4 or 5 more minutes and then I can collect that one. Once I store it away, I just gotta keep checking the temperatures and everything on my tubes to make sure that everything is okay and sterile. And you do have to pass the tubes over the flame, the lip of the tube by the flames to sterilize it prior to using the pipette and pouring it into the test tube. That's a wrap for my first part of the experiment. I will film again for the second part once I can show the growths and then put it on the petri dishes. My name is Marley Romero, Microbiology, Week 2, Exercise 2. And this video is for Exercise 2 for Microbiology and 3. So I'm gonna show you, I wiped down my table with like a bleach spray and then bleach wipes. Then I set down like the Chuck's absorbent pads in case I spill anything. Let me see if I can flip the camera. Okay, I can't. So here are my labeled tubes that I'm gonna use for the separation into the colonies. And then I have my tubes with my growths here. I have alcohol and I'm sterilizing my illocation tube loops. The pipette, I have distilled water, my yeast ready to go. I have all my little petri dishes labeled. So here I have the agar boiling so that it melts. And then I have here the coffee mug where I'm gonna put them in hot water still so that I can be able to pour. And I have my little oven mitt thing so I can get them out. Okay, so I now put my cultures inside my poured agar petri dishes. And according to the instructions, I have to let these sit for another 48 hours. So I'll have to go back and check and keep tabs and see if anything grew. And then right now I am letting my yeast activate so I can separate into multiple colonies so I can see what actually grows. And then from there I have to pipette it into the tubes with distilled water. Then after, put a little bit of yeast into the first one, and from that first one transfer it to the second, then from the second to the third, and so forth. And that's how you dilute the microorganisms. And then once they're poured into their own petri dish, it's gonna sit there for 12 hours. After the 12 hours, I will go check and see what I found and how many colonies I can find. And I have my stuff here, my candle, because you have to sterilize every time you open your culture. You have to sterilize the lid, the lip of the vial every time, and then when you open it and then right before you close it, just to prevent any other organisms from entering your culture. And I guess I will check back in 48 hours and I can complete my assignment.